All right. So we're going to continue here with sort of the second overview of microbial diversity before continuing to talk in more detail about specific aspects in the next few lectures. So the next lecture later today will be focused on what you could call stealing functions. How organisms acquire functions from other species, either by taking their genes or by some type of symbiotic interaction with other organisms. So I'm just going to, we covered this right at the end last time. I'm going to cover just really quickly this issue of the different forms of trophy, this function wrap-up, and then we're going to however quickly I can, march through the phylogeny of everything else in the tree of life other than plants, animals, and fungi. A lot of diversity to cover there, and I'll try and tell you what aspects you might need to know about and remember versus what is sort of just to introduce you to the topic. So the reading for this, again, is chapters 26 and 27. This covers the diversity as I've presented it, in essence, the diversity of form, some of the diversity of things like motility that I already talked about, the diversity of function, and then the phylogenetic diversity in 26 of bacteria and archaea, and in 27 of microbial eukaryotes, really everything but plants, animals, and fungi. And you really have to read chapters 26 and 27 and understand much of the material in there, and I'm going to try and give you some guidance about what aspects of it to focus on as we talk through it. In addition, I will send around an email over the weekend. We're going to schedule some review sessions for next week for the midterm, and hopefully over the weekend I'm going to send out at least some practice questions, if not a sort of demo practice test that will try and be about the same length as the real test. I'm going to try and get that together maybe by Sunday for people, but then we're going to try and schedule a couple of review sessions where you can come in and ask whatever you want to ask in those sessions for next week. So again, reminder, we were talking about these forms of how organisms acquire their nutrition, talking about this in terms of the trophy status of organisms, and I showed you these 3 ways to categorize the nutritional status of organisms. We can divide it up by their energy source, by their electron source, and by their carbon source. And I already sort of introduced this and went through a couple of examples. There are lots of different ways you can categorize the nutrition of organisms. These 3 that I focus on here are the 3 most commonly used and 3 of the ones used in the book. So when it talks about different lineages, it might say that this group of organisms is blottity trophic. That's why I want to go through this so you understand what it's saying when it talks about those different organisms. So again, if they use the energy of light, they're called phototrophic. If they use the energy of chemicals, they're called chemotrophic. If they use electrons from inorganic sources, they're lithotrophic. From organic sources, they're organotrophic. And then if their carbon comes from C1 compounds, they're autotrophic. And if it comes from organic compounds, they're heterotrophic. And I think I stopped somewhere around here and gave you a couple of examples of this. So E. coli, classic model organism studied in labs all around the world. It also happens to live in our gut and some of the time makes us sick. There are certain strains of E. coli that make us sick. E. coli is a chemo-organo-heterotroph. Basically uses sugars, for example, for everything. And so it uses chemical energy from the sugars. It uses the sugars as its electron source, and it uses the sugars for carbon, for example. It can also live off of other organic compounds, but sugars would be one of them. So it's chemo-organo-heterotrophic. Sometimes that's just called chemoheterotrophy. And the other example that I introduced was cyanobacteria. They're photolitho-autotrophic. So are plants generally, and many of the things that you would call photosynthetic. Photosynthetic is sort of another word for photoautotrophic. You could say the whole thing, photolithoautotrophic, but usually people just say photoautotrophic. And humans are like E. coli, chemo, organo, heterotrophic. Yeah, question?
So why do people usually leave out the name from the electron source?
because electron source is sort of an obscure thing that most people don't want to think about, I think. And it's less sort of related to a lot of the things that people are interested in organismal biology. So autotrophic, whether or not something is pulling carbon out of the air, for example, is very important for the trophic sort of status of an organism, whether it's, the primary producer or, consuming things. The exact source of its electrons is a little more esoteric for a lot of other questions. So people frequently, the things they care about are they getting their energy from the sun or from chemicals, and are they getting their carbon from C1 compounds, that is usually gases like methane or carbon dioxide or carbon monoxide, or are they eating other things? So I think that's probably why. And in the book and in many other places, they don't even use frequently they'll just say something's photoautotrophic, for example. So if you look at that table that I showed you, or partition any other aspect of metabolism of organisms, you see something sort of you can generally apply across the tree of life, which is that in terms of biochemical diversity, the diversity of manipulating different compounds, breaking bonds, stitching together various bonds, bacteria and archaea broadly across all the bacteria and archaea are incredibly diverse. Individual species may not be that diverse, but if you sum up all the bacteria, there's an enormous amount of diversity there. Even if you look within one of these categories, so for example, phototrophy, the bacteria that are phototrophic, there's incredible diversity within that category. So for example, for photoautotrophic, there are at least 6 distinct major groups of bacteria that appear to have, in essence, separately invented or separately developed some parts of photoautotrophy. And they don't do it all in the same way. Some of them have similarities in how they harvest the energy of light, but many of them fix carbon, for example, in very different ways from each other. So even within one of these categories, there is enormous diversity in bacteria and archaea, and there tends to be somewhat less across eukaryotes, especially in the multicellular eukaryotes. The diversity of metabolism there is lower, and that's one of the reasons, most likely, why eukaryotes in general and multicellular eukaryotes in addition frequently engage in some type of symbiosis with other organisms in order to acquire the functions that they need. So degrading cellulose or fixing nitrogen or fixing carbon. Most of those things eukaryotes don't actually do on their own. They have some, usually bacterial symbiont, occasionally an archaeal symbiont that carries out those processes for them. There are some fungi and some microbial eukaryotes that have diverse metabolic properties, but it's on average much lower. So what I'm going to do now, this will be a little, might hurt your brains a little bit, but we're going to sort of walk through the diversity of these different lineages. And it's important to understand some of this diversity. I'm going to try and tell you what aspects of that phylogenetic diversity we're going to expect you to be able to regurgitate or know when you go in for the test. But in order to talk about things like evolution of photosynthesis, which we will talk about, symbioses, extremophiles, gene transfer, human-associated microbes, we have to introduce you to these different lineages because some of them are involved in all these different processes that we're going to talk about. And the tree itself is important for understanding many of these different processes. So if there are no other questions about the sort of functional diversity, I'm going to move on into our little march through the tree of life here. Okay, so we're going to use the rooted tree of life as our guide, these 3 main branches in the rooted tree of life. And just since we haven't talked about this in detail, and I'm not going to talk about this a lot, when we show phylogeny for particular groups, say the phylogeny within the microbial eukaryotes or the phylogeny of bacteria or of archaea, a lot of this is based upon molecular data, like the ribosomal RNA data that we talked about for finding the 3 domains. So the relationships within the 3 domains a lot of that is based upon molecular data. In particular, for bacteria and archaea, this is true. For microbial eukaryotes, for many of them, there are morphological and other features that are actually very informative for phylogenetic analysis, and I will show you some examples of this today. But for bacteria and archaea, in general, appearance, the morphology of them, just as it was not useful for discovering this third branch in the tree, it is also not useful for sub-classification within each of the branches. And so historically, there have been an enormous number of misclassifications of things that we now know as bacteria or archaea as something else. Sometimes as viruses, sometimes as fungi, all sorts of, now in retrospect, crazy classifications of organisms. When molecular data came along, it led to a restructuring of the entire sort of bacterial and archaeal tree. What I'm going to present to you here is sort of the more modern view of the bacterial and archaeal tree. And the microbial eukaryotes, much of that is based upon sort of a mix of molecular data and morphological data. And then as you get into fungi and plants and animals, you will hear a little bit about what some of that is based upon. But when you have lots more morphological features or fossil record, you can do things other than use sequence data. But for bacteria and archaea, the morphology is just not informative, generally. So in general, for bacteria and archaea, you should remember the major groups we're going to talk about. There are going to be 6 of them for bacteria and 2 of them for archaea. You should be able to give examples of some of the properties of those groups. And in addition, when we talk about evolution of features, you should understand how the features map onto the tree. We'll talk more in detail about what you're going to have to understand about the 18 or so microbial eukaryotic groups, which I can't even remember all the names all the time, so I'm not going to ask you to memorize all those either. So we're going to go through the bacteria first. If you look at the tree in the book, there are 6 major clades of bacteria represented in that tree. The exact topology of this tree is, they probably should have used something else. Many people right now think that, for example, the groups called low GC gram-positives and high GC gram-positives are probably sister taxa and should be next to each other in the tree. As I mentioned before, that suggests that gram-positiveness evolved once in the phylogeny of bacteria. So the exact topology of this tree is to be taken with a large grain of salt. But we're going to focus not on that topology, but on the individual groups here. These 6 clades are examples of 6 of the some 100 major lineages of bacteria. We are not going to go through those 100 lineages. These are the 6 sort of best understood and best studied, and those are the ones we're going to focus on here. So the first one we're going to talk about is the Spirochetes. These are going to be familiar to people mostly for the ones that cause diseases. So the causative agent of syphilis is in this group. The causative agent of Lyme disease is in this group. They are, as the name suggests, spiral-shaped. So they have these sort of corkscrew shapes to the cells. And a very interesting thing that they do, the flagellum that many species use on the outside of the cell to swim around in the water, they embed, in essence, a flagellum inside the cell, and then they rotate that flagellum-like structure around, which creates a screw-like motion, which is very unpleasant if you are infected with one of these. That's how they burrow into things, by spinning their flagellum that's inside the cell and creating this sort of corkscrew-like motion. They're all gram-negative. That is, they stain negatively with the gram stain, and they have that double membrane structure that I showed you. Many of them are motile with this sort of weird axial filament. So the drawing down here is, that's sort of the flagellum inside the cell. The ones that are best known and best studied are human or other pathogens, but there are some free-living organisms in the group, live in all sorts of interesting places like acid springs and soil, etc. They're just not as well studied or understood. So one other interesting thing about the spirochetes, the one that causes Lyme disease, Borrelia burgdorferi, although most bacteria have circular chromosomes, it has linear chromosomes. And, well, actually linear large pieces of DNA that you could call plasmids. And those, the ends of those are hard to replicate, just like the ends of linear chromosomes in eukaryotes, and it uses a telomere-like process just like eukaryotes use, invented completely separately, but it carries out the same general function as the telomerases and telomeres in eukaryotes. So this is the second major group of bacteria, the chlamydias. These, I don't think as of yet any representative of this group has ever been found to be free-living. They are all known, currently at least, as obligate parasites living inside cells of eukaryotes. Many of them are known to be what are called energy parasites. They actually can't make their own ATP. They steal ATP from their host. That may explain why they don't have a free-living phase. It's hard to live when you can't generate any of your own energy. They are all gram-negative. They actually have diverse shapes. A very strange thing in their classification, if you got, a long time ago, one of these diseases that are caused by these things, like trachoma, which causes blindness, or the sexually transmitted diseases that are named after the organisms in this group, or pneumonia, which is also caused by this group, people would think you were infected by multiple things because these organisms can actually take many distinct forms. So one species can be both rod-shaped and sphere-shaped, and sometimes even form long filaments versus clusters, and people thought you might have an infection with many things when it was really one organism with many different forms. They're also very, very small. So this is an example of one of the groups that many of them were misclassified actually as viruses initially, before molecular work showed that they had all the molecular underpinnings of bacteria, and then ribosomal RNA trees showed where they sit within the bacterial tree. So the third group of bacteria I'm going to include in this tour are called the Actinobacteria. That's the sort of geeky microbiologist name for them. The more common name is the high GC gram-positives. And what that refers to is that these stain positive with the gram stain. They have that structure to the cell envelope that I showed you before with the peptidoglycan, thick peptidoglycan cell wall, and only one membrane around the cell. And their genome, the DNA in their genome, has a lot of G's and C's in the genome. So DNA is double-stranded. There are the base pairing rules, A with T, C with G. So whenever an organism puts C on one strand of its genome, it has to put G on the other strand. So you can sum up the total number of A's and T's together in a genome and the total number of C's and G's. And the percentage of the genome that is C's and G's is sometimes a useful diagnostic. This group of organisms, all of them have high frequencies of C and G in their genome. It's unclear if there's any functional significance to that, but it is sort of a diagnostic feature, and that's where they get the name. Many of them form these elaborate sort of branching structures that look a lot like elaborate branching structures of fungi. Many of them were misclassified as fungi prior to molecular work. And that's kind of where the Actinobacteria name comes from. Actino is also frequently used to describe some fungal groups. Many of them even form spores at their tips and look a lot phenotypically like fungi. A lot of the antibiotics that we use come from members of this group. So streptomycin is from Streptomyces species. Many of these are soil-living bacteria that make drugs or chemicals that manipulate and or kill other microbes in their environment. And many of them are very useful as sources for antimicrobial compounds. And a lot of the fungal and bacterial antibiotics have been isolated from this group. There are many pathogens in this group too, like the causative agent of tuberculosis and leprosy. So group number 4 is going to be really important for many of the other things we talk about later today and in a few other lectures. These are the cyanobacteria. They used to be known as the blue-green algae. but they are phylogenetically not algae, not eukaryotes. They are bacteria. So they're formally known as the Cyanobacteria. Almost every member of this group that has been studied is photolithoautotrophic. They use a particular pigment called chlorophyll A as part of their photosynthesis. They also generate oxygen as part of their photosynthesis and have been really important in producing the oxygen in the atmosphere on the planet a few billion, some billion years ago. They are also the closest relatives to the chloroplasts that are found in algae and plants, and we will talk about that later today. Many of them form these long filaments. A lot of them fix nitrogen and do interesting things and are symbiotic, for example, with fungi or other organisms to help them acquire nitrogen. Some of them are spiral-shaped, so this is a spiral-shaped one of them that also does this sort of corkscrew type movement. I don't know if it does it in the same way as the spirochetes or not, but it's sort of, there are many spiral-shaped organisms across the tree of life. I think this is actually spirulina. If you get some of the green food drinks, sometimes they use one of these cyanobacteria as the protein and the green in your drink.
This sequence shows the gliding corkscrew movement of the cyanobacterium spirulina.
Turn down the lights a tiny bit because I have a couple of little videos at some point. So another group is formerly known as the Firmicutes or Firmicutes, some of us call them. They are gram-positives also. Their genome G+C content is low percentage, on the order of 30 to 40%. So they're also known as the low GC gram-positives. There are a couple of lineages within this group that do not have the gram-positive structure. They have in fact thrown away their cell walls. So there's a group called mycoplasmas that cause all sorts of nasty diseases in people and plants and animals that don't have a cell wall anymore. There are many pathogens in this group that do have the gram-positive cell wall, so Staphylococcus, Streptococcus, etc., causative agent of botulism and tetanus. A lot of them, I think I gave an example of this before, a lot of them form this spore structure inside the cell, so they can, instead of splitting the cell down the middle like many bacteria and archaea do, they actually have a daughter cell produced inside the mother cell. They coat it with all sorts of protective coatings, and then the mother cell dies and disappears and the spore is left. And those spores can survive for thousands, if not tens of thousands of years, and then germinate when they get exposed to water, which you really don't want to happen if, for example, you inhale anthrax spores into your lung. And that's why they germinate. They germinate when they get exposed to water. And if you got 5 or 6 of them in your lungs, you're dead. So the sporulation process is really interesting, and we'll talk about that in a little more detail in another lecture. So the last group of bacteria I want to tell you about is another really important one for a few of the other lectures we're going to talk about. This is the Proteobacteria. In terms of number of species that have been characterized, as well as some other aspects of diversity, this is the largest group. The most species that have been experimentally characterized of bacteria are in this group. Incredible diversity of metabolic properties. There are photoautotrophic members of this group, chemoautotrophic members of this group, obligate intracellular pathogens, incredible diversity within this group. E. coli, the model organism for all sorts of molecular biology, is in this group, as are many pathogens like the causative agent of plague and cholera and various other issues. They're all gram-negative. The mitochondria is derived from within this group. So when we talk about mitochondrial evolution, this group will become a little more important. So that's your tour of the bacteria there. So now we're going to switch over to talking a little bit about the archaea. Fortunately for you guys, there's a little bit less diversity known in the archaea. In general, when people have characterized the archaea, there seem to be fewer major lineages compared to the bacteria. It may be because they're more recently separated as a lineage. It's not entirely clear. There are only 2 major lineages of the archaea that we're going to go over and that are also covered in the book. There are more additional lineages. We're just going to cover these 2. These are the Kren Archaeota and the Euryarchaeota. The Cranarchaeota, many of the members of that group, if not most of them, are what are called thermophiles. They like to grow at high temperatures. We will talk more about this in the lecture on extremophiles. Many of those are also acidophilic, that is, they like to grow in high acid, also low pH. High acid is low pH. So for example, one of them, which is the best studied in the group, probably Sulfolobus, grows at about 70 to 75 degrees Celsius. That's a temperature higher than any eukaryote sort of is able to thrive at. So you basically, when you cross over 70 degrees or so Celsius, you tend to only see bacteria and archaea dominating those ecosystems in a pH of 2 to 3, so sort of warm, very hot acid. Others grow at pH 0, that is like pure sulfuric or hydrochloric acid. And some of them grow at temperatures above 100 degrees Celsius. Basically, anywhere where water is liquid, there seem to be archaea that have been able to survive in those environments and thrive. So in the bottom of the ocean, where pressure compresses the water and makes the boiling point higher than 100 degrees Celsius, there are archaea that thrive in those conditions where there's like a volcano, and some of them have been shown to have optimal temperatures of growth at 110 degrees Celsius. Kind of completely ludicrous, actually, and how they're able to keep the cell together in those conditions is still not very well understood. So the second major lineage of archaea are the Euryarchaeota. There are two main phenotypic groups within the Euryarchaeota. One are the Methanogens. These are organisms, as the name suggests, that generate methane as a byproduct of their metabolism. They are responsible for an enormous contribution to greenhouse gases on the planet. Methane is a very potent greenhouse gas, and these methanogens, for example, live in the guts of probably almost all ruminants on the planet, like cows, and when cows burp or something comes out the other side, methane frequently comes out, and that's coming from these methanogens. There are methanogens in the gut of people as well. Not everybody, but many people have methanogens in their guts, and exactly what they're doing is not very well understood. Methanogens are not a monophyletic group, however. If you map their presence onto the tree of the Euryarchaeota, they're scattered throughout that tree. And there actually are, I believe, some Cranarchaeota that are also methanogenic. So methanogenesis is scattered throughout the phylogeny. There's another group of Euryarchaeota, however, that is monophyletic. So one subgroup of Euryarchaeota is able to thrive and grow in very high salt concentrations. These are the halophiles. There are many halophiles of all sorts of different organisms, but as you get to higher and higher salt concentrations, what you're left with is basically only extremely halophilic archaea. This one clade of archaea is able to survive and thrive basically in salt concentrations where the salt is literally precipitating out of solution. And they are able to maintain their cell structure in the face of this. And we will talk a little bit about this, how they do that in the lecture on extremophiles also. Many of them have these pink carotenoid pigments, and most of them also have a really interesting protein. It was originally called bacteriorhodopsin before people knew about archaea as a third branch. They haven't renamed the protein yet. It's still called bacteriorhodopsin. And it's like the rhodopsins in our eyes that allow us to sense light. It's not appear to be evolutionarily derived from the same common ancestor, a separate sort of invention of this process. But they allow these halophiles to take the energy of light and create a proton gradient between the inside and outside of their cell. They pump protons using the energy of light. They can take that proton gradient and then make ATP, make cellular energy. So these are phototrophs, but they do not fix carbon. They do not live off of C1 compounds. So they are photoheterotrophs, not photoautotrophs. So they're using the energy of light for part of their biology, but they are not using it
like cyanobacteria. Yeah.
So did you say that bacteria rhodopsin is a type of pigment?
It is pigmented, actually. It does, so just like the rhodopsin in our eyes, it binds to a cofactor that helps it absorb the energy of light, and that makes it reflect certain colors. So in essence, it is a pigment. These organisms also have another pigment that I wrote about on the top here called carotenoids that help make them pink. So they're very sort of colorful organisms. This rhodopsin absorbs the energy of light, and what wavelength that it absorbs at is dependent upon this molecule that sits inside of it called retinal. And that's very similar to what we do in our eyes in many ways. So more importantly, they use that. Some of these pigments they just use for sort of whatever, not entirely clear what some of the carotenoids are used for. These are used to generate cellular energy. Yeah?
Would they be able to survive in like pH 7? Or they don't live salty?
So salt is not that. So the pH can be independent of the salt concentrations because the pH is the concentration of hydrogen ions. And many of them can live, so some of them can live in very basic conditions that are also very high salt. So you can have Mono Lake, for example, up at the top above Yosemite, is very high pH and also very high just total salt concentration. There are also very low pH environments that are also very high total salt concentrations. They're all throughout there, but they cannot live if the total salt concentration is low.
Okay.
So they are obligate for the salt being very, very high. If you lower the salt, they actually pop. As we will talk about in the lecture on extremophiles, they can't maintain their osmotic pressure inside the cell. If you put them in lower salt concentrations, as I did at the beginning of my PhD, not knowing this, because I studied them, and I killed them all. It was fun. Someone had sent them to me from Canada, and took a month and a half to grow them, and then I killed them all. Yeah. Didn't hurt. So that's our mini tour of the bacteria and archaea. And again, I'm trying to set this up because we're going to talk about many of these different lineages in a variety of different ways for the next few lectures. So now we're going to do what may be a bit more painful, which is a tour of the microbial eukaryotes. And probably in part because microbial eukaryotes have many more morphological features, and many more of them actually form larger structures, like multicellular structures, like the giant kelp I mentioned before. There are many more groups that are named that are studied in detail. And so bear with me here. We're going to go through some of these. You are not going to have to memorize every detail of every single one of these groups. In the second lecture today, I will tell you a little bit more about which parts of the biology of these organisms and their features you should remember more of and which you should understand. But I want to give you this introduction because in these 15 minutes is sort of covering a lot of the diversity of these other lineages that are really important for understanding things like evolution of plants, evolution of animals, and evolution of features like chloroplasts and mitochondria. And again, as I mentioned, the phylogeny that we think we know for these groups is largely based on a combination of molecular and morphological and other data. So if we look at the eukaryotic branch in the tree, here's a figure from the book. Yeah, well, get ready. As I said, you're not going to have to memorize all the details of all of these lineages. I don't even know all the details of all of these lineages. However, they're really interesting and really important, and there's incredible diversity here, and it is going to be very relevant to the things we're going to talk about, so I want to give you a tour for a few minutes. If you look at this tree, note that there are polytomies in this tree. There are also these dashed lines in the tree. In essence, you can view this tree as being ambiguous for everything below the common ancestor of each of these 5 major groups. So you should focus primarily on these 5 major groups here. This one here, this one here, this one here, this one here, and this one here. Those are the big lineages of eukaryotes we're going to spend a lot of time talking about. I'm going to go through the details of some of the subgroups because they're interesting and important, but the thing you should focus on a lot of are those 5 big groups. In particular, one thing to look at is whether or not lineages have a mitochondrion. Mitochondrion is what we use to generate ATP energy from glycolysis. It's a very important part of the eukaryotic cell. We will talk about this more in detail in the second lecture. And whether or not organisms have a plastid or a chloroplast. And this is very important for photosynthesis in many different lineages. Okay, so big giant group number 1 is the chrome alveolates. The name sort of comes from a combination of 2 different lineages. One that were called the I don't even know the original name, and the alveolates, as we will see. And they've been merged together into one giant group that people think may share a common ancestry with each other. If you look within this lineage, there are 7 different major subgroups within the chrome alveolates. Each of them are important and interesting in and of their own, and we're going to quickly go through them. There's a nice table in the book that summarizes sort of a major feature of each of these lineages that I'm going to go through today. It's table 27.1. It's a very long table. So for many of these groups, and this is one thing you might want to focus on, for many of these larger groups even, or some of the sort of subgroups like the alveolates here, there are features that they all share in common that appear to be synapomorphies of that clade. So this tree is largely based upon molecular data, but the molecular data is consistent with many synapomorphies that have been observed for features in these groups. So the alveolates have at least a few synapomorphies. As the name suggests, one of them is the presence of these alveoli, which are basically sacs beneath the surface of the plasma membrane. If you look within the alveolates, there are 3 distinct groups. You don't have to worry about the exact details of each of these groups, but you should know some of the major features across eukaryotic evolution. One of them is the apicomplexans. This includes the causative agent of malaria, as well as toxoplasma and a variety of other, I think all of them are obligate pathogens. They do have a synapomorphy they all share with each other, and we will see in a little bit that they also all have plastids, which is very unusual because none of them are photosynthetic. The life cycle of Plasmodium, which has gone through in the book, is really pretty interesting. They infect things like people in their blood and are transmitted by mosquitoes, but the life cycle inside the mosquito is an obligate part of the sexual life cycle of these organisms. And if you can screw with that part of their life cycle, you can help reduce the transmission of malaria, for example. Second major group in the alveolates are the dinoflagellates. The name refers to a synapomorphy for this group, which is these 2 flagella, that if you look at this little video here, that's how some of them move around in the lower right-hand corner. They have these 2 flagella that they spin around, and they can control them in various ways to guide them in various directions. As you will see later in the class, dinoflagellates, one of the interesting things about them is that they are symbionts of coral. So the photosynthetic parts of many coral are dinoflagellates. If you have bleaching of coral, it's partly because they've lost their symbionts. Dinoflagellates are also the cause of many of the toxic red tides that you see in various coastal regions. They're also responsible for a lot of the photosynthesis on the planet. The third group in the alveolates are the ciliates, like one that you may or may not see in Lab 3, ciliates from the gut of termites. Here's the paramecium ciliate, which many people may be familiar with sort of introductory labs. They all are coated with cilia, and those cilia are used for a variety of processes. There are some very important model organisms in this group. So Tetrahymena, a model ciliate, has been studied in detail in the laboratories of various scientists, and work in Tetrahymena has led to something like 6 or 7 Nobel Prizes. So that's where telomeres were discovered. That's where various parts of histone chromatin structure was discovered. That's where catalytic RNA, which you may have heard of, was discovered, and a variety of other interesting things in studies of ciliates. Most of them are poorly studied. There's incredible diversity within the group. New species being found basically whenever someone goes out to look for them. Very high levels of diversity in a lot of environments and very poorly understood.
This is one of the many bizarre
So a second
organisms
major
found
group
in the gut of
within
terrarium prey.
the
It is a very
alveolites,
large,
the
single
name
cell.
that,
The area show chlamydomonas within this being ingested in for feeding
Halophiles,
synchronized, which is flagelled off at the
but
base
this
of the
group
gullet
here,
and moved
they do have
into the
many
cell.
features in common. There appears to be a synapomorphy of this larger group in terms of this dimorphism that they have with their flagella. If you look at them in the microscope, you can see this. This is partly why they were grouped together, but it's supported by molecular data. This group includes brown algae. All of the brown algae are, as far as I know, or almost all of them are marine and multicellular. This includes the giant kelp of kelp forests, very important ecologically and very interesting from a lot of evolutionary points of view, a separate invention of multicellularity compared to the multicellularity in plants or animals or fungi. This whole lineage is basically multicellular, yet it's distinct from basically like brown plants in some way. Diatoms are a separate lineage within this group. They carry out a lot of the photosynthesis on the planet. We know an extraordinary amount about the evolution of diatoms because they form excellent fossils. They form these hard outer casings to the cell, and there's just incredible fossil record of diatoms that has allowed us to understand their evolution better than many other microbial eukaryotes. Many of them are also photoautotrophic. Nasty group of organisms, in my opinion, are the oomycetes. Like the Actinobacteria, they were originally classified as fungi. They are not closely related to fungi, but they look like them. Molecular data reclassified them. That's really important for this group because, for example, they include the causative agent of the potato late blight, the potato famine. If you want to try and treat plants infected with these oomycetes with antifungal drugs, they basically don't work because they're not fungi. They are unfortunately also the cause or one of the causes of the
some of the
sudden oak
largest
death going
organisms
around
on
in
the
the West
planet,
Coast.
specifically the
They're pretty
giant
nasty
kelps of the
organisms
marine
and very
kelp
hard
forest.
to get rid of. So the last group within the chromalveolates are the haptophytes. These are just beginning to be studied in more detail. People have been known about them in many ways, sort of because some of them form these complex cellular coatings, but we haven't known a lot about their biology. One of them is this Emiliana huxleyi. It is smaller in size than many or most bacteria. Was ignored by a lot of people because they just thought it must be a bacteria and not a microbial eukaryote, but it probably contributes to, that one organism contributes up to 5% of the total photosynthesis in some places of the ocean. This, for example, is a bloom probably of that one species, imaged by a satellite.
This is a colonial species of diatom that displays
So
a
that's
highly
one of the
specialized
5 groups.
gliding motility, shown here
A
speeded
second
up
group,
5
which you will
times.
hear a lot more
The
about, and
cells are
I'm really
linking
going to
by
just
a
quickly
tongue
cover
and
now,
groove
because I'll
system
talk about
along
them
their
later
silica
today a little
wall.
bit, and you will hear more
Movement
about
is
plants
generated
later,
by
are the
an actin
plantae.
system coordinated in the colony.
This lineage here, it includes 5 major subgroups. One of those subgroups are the land plants that you will hear about in 2 weeks or so from Brian. All of the members of this group basically are photoautotrophic, at least their common ancestor was. Occasionally there are parasitic members of these groups that have lost their ability for photosynthesis. Again, the table has some detail about them. One really interesting thing is, for example, the glycophytes, this first lineage to branch off, their chloroplast has peptidoglycan in it. We will talk about that more in the second lecture today. The red algae, as their name suggests, are mostly red in color. Incredible diversity within this group. One of the things that people study a lot of in this group is their mechanisms of sporulation and sexual reproduction. but some sort of incredible diversity. There are both single cell forms and multicellular forms within this group. The chlorophytes, we will talk about also in a little more detail. There are mostly green, sort of the chloro part of the name implies that. Some of them are multicellular, some of them are single-celled. We're not going to talk in detail today about, you're going to hear more about land plants and charophytes, I guess, from Brian later. I might talk about charophytes a little bit. And we're going to talk about that lineage later today when we talk about chloroplasts. So lineage number 3, you ready? The excavates. This is a list of the 5 major lineages of excavates with some of their features. One of the most interesting and important things that we're going to talk about today about this group is that 2 of the lineages, the diplomonads and the parabasalids, do not have mitochondria. Single-celled organisms, they are eukaryotes, they have a nucleus, but they do not have a mitochondria anywhere inside of them. It includes many parasites within this group. There are some free-living members of this group, I think, but none have yet been found to have a mitochondria. That will come back later today when we talk about evolution of mitochondria.
This sequence shows the Fall
Third group, which
actively
is,
swim
I
the
think, very
motility
poorly studied, except for
synchronized
this one
Magellan
which is the
in which small
heterolobosians.
currents and is
One of
able
them
to
is
propel the
this
colony.
nasty thing called Naegleria, which causes these brain infections. You do not want to get Naegleria. 2 other lineages within this group will come up again later today. The euglenids includes a model organism called Euglena, which is a photosynthetic single-celled organism, and we will see has some unusual parts of its photosynthetic history. And then the kinetoplastids, most of them are obligate parasites. It includes things like the causative agent of sleeping sickness, leishmaniasis, and Chagas disease. They do have this synapomorphy in the group. That is the presence of this kinetoplast, which is actually sort of, in essence, a component of the mitochondria in these organisms. So group number 4 in this giant collection of organisms is the Rhizaria. Fortunately, it's a smaller group in terms of number of lineages. There are 3 major lineages of Rhizaria. They do have what appears to be a synapomorphy, which are these long, thin pseudopods. The Cercozoans are very poorly studied. You'll see a little bit about them when we talk about the evolution of photosynthesis, and I'm going to end today with just talking about these foraminiferans, which, like the diatoms, although in a separate group, have an excellent fossil record. There's some great research on the evolutionary history of foraminiferans. As I think I mentioned in this class before, there's one of them that lives in the deep sea that is the size of your fist, and it's a single cell.
This
And we
real-time
will end with, you can watch this little video from the cruise where we collected that actual organism. So if you search for the name of this organism, you pull up that picture on Wikipedia. It came from this deep sea dive off of the Galapagos Islands. And, oh no, I've killed it. I'm just going to leave it running. In about a minute, you will see, and I can show you this in the second lecture too, you will see the submarine approach this giant single-celled organism. And I'll see you later today. I had to do the tour.
So how did you go down on Alveolites?
I didn't go down.
Oh, you didn't?
I went on the cruise, but they made it, you know, as a diabetic, it was going to be complicated to go down. I'd have to get medical approval. And
I want to ask, how important is the new edition of the book? Because right now I'm using the old edition.
chapters 26 and 27 appear to be almost the same.
Okay.
I don't know about the other chapters,
Okay.
so I can't say.
And I don't know my section because I only had.
As far as I can tell, you won't see anything really different in 26 and 27. That's a Brisingid star. I have that in the freezer.
Wow. That makes sense.
Yeah. And then you'll see them come up to the giant Xenophyophore. There it is. No, not yet, not quite yet. They're going to come around the corner. There's a anemone. This is like 2 miles. So I have this giant thing in my freezer over in the Genome Center, by the way. It's a freezer filled with tube worms, and there it is. Before they grabbed it and killed it. It's a cool story because nobody knows much about these, but there was a graduate student in the Alvin submarine when the submarine went down, and she, as part of her first year graduate project, had made a catalog of all organisms that had ever been observed in deep sea dives. She was basically the world expert in the diversity of the deep sea. And they're cruising around, and the Alvin pilot, is cruising around in the bottom of the ocean, and one of the chief scientists on the cruise, they saw that thing and they just ignored it. They had no idea what it was. And she's like, no, stop! That's a Xenophyophore. That's a single-celled organism. And there are like 2 samples of them. But only because she had been making this catalog did they collect it. It's completely cool.